Okay, ladies and gentlemen, slowly but steadily, I think we should be starting. It's 1:45 now. We have a very full program. We have a really exciting program. What we try to do this year is really to move towards translation, looking at, in particular, also medical doctors. We have 2 or 3 in the lineup, and companies that work on clinical products, to really see where the nanomedicine field, in combination with imaging, so the theranostic field, where that will be going in the next couple of years. First lecture will be given by Daan, formerly the head of the Department of Pharmaceutics at Utrecht University. We've been discussing quite intensively over the last year or so on where to go with Nano, right? So what things are needed for the future, what is the current state, what topics should be considered. And I'm very much looking forward, Daan, to hear from you where you think we all should be going in the next couple of years.
Well, thanks, Twan. First of all, they kicked me out of the university, so I had to find something else to do, and I can look around, I can reflect on things, and talk with young, enthusiastic scientists like Twan, and be very optimistic about what is going on. But today I feel a little bit like well, one of these grumpy old man who give commands to what's going on the podium while they're sitting somewhere in the side wings. I think I'm the left one, isn't it? Yes. I'm not going to tell you who is on the right side, but he's sitting somewhere there. Okay, on the menu, 3 things that I would like to sort of, like I said, reflect upon. First, annus horribilis and beyond. The previous speaker, Klaus Michelier, also used some Latin, so I thought I should throw that in as well. What are animals trying to tell us, and molecular antibodies banned from this conference, but for what reason? And because they are normally not seen as nanomedicines. First of the 3, annus horribilis 2016. Bind collapses, and then we got this paper by Stefan Wilhelm and Warren Chan, where the bottom line was, well, there's not a bottom line here. Yeah, there's a bottom line here. Only 0.7% of the administered nanoparticle dose is found to be delivered to a solid tumor. And that is, of course, when you read that as an outsider, that is bad news, because all these nano people trying to target and have big mouth about targeting, and what is the result? 0.7%. So we got reactions, the more emotional ones, why I'm happy holding on to hope for nano in oncology, and the more strict and straightforward, Scott is sitting somewhere there. Evaluation of nanomedicine, stick to the basics, in other words, shut up. And we, of course, being Dutch, being very nuanced and always trying to come up with the consensus, a compromise. We actually spent, and I'm talking about we, Twan and Gert Storm, myself, and Fabian, we were spent our summer thinking about that. And what Wilhelm is just, the mistake he's making is that he's talking about technology and about science, but basically what you have to do is you have to talk about the patient. What counts, what counts is patient benefit. This is largely independent of the percentage of the injected dose that can be delivered to the tumors, but instead results of a shift in balance between off and on-target accumulation. So that's what counts, and nothing else. And that's why it's so important that CLINAM, the CLI from CLINAM, the clinical part, is emphasized, should be emphasized, and that's why this conference is so important for the field. So this is the first part. Annus horribilis 2017 gives us new chances and a better view of what we really should be aiming for. And that is not targeting per se, that is treating patients. So I will move now to the next one. That was about animals, but if you look at this slide, the first thing I would like to point out is that when you're talking about drug targeting, the best lessons you can draw is always from Mother Nature. And so there are very impressive targeting approaches Mother Nature developed, like iodine and the thyroid. Almost 100%, it's a little bit more complicated, but almost 100% ends up in the thyroid and nothing else elsewhere. That is the simple story. Now, Jesse is sitting here, and he will remember this slide very well. These were the first slides on liposomes and on the EPR effect, the enhanced permeability retention effect. And these very dramatic pictures came up for patients, of a patient with Kaposi sarcoma. And it's very clear that the enhanced permeability and retention effect, that it works in a number of patients, in certain patients. And later on, the question is, of course, who's the candidate? And you will talk about it as well, of course. So impressive accumulation, successful. Kaposi sarcoma sort of disappeared when the AIDS epidemic disappeared, was treatment improved. So Kaposi sarcoma also became less prevalent. So to some extent, targeting was observed, had been observed when Maeda in Japan came up with this EPR, this enhanced permeability retention effect. And so everybody sort of jumped on that and started working with animals, and that's where my second point comes in. Animals, what are the animals trying to tell us? And we really trust and rely so much on animals that I think a little bit of caution should be exercised here. And there is also quite a bit of literature about that. When I was director of the Top Institute Pharma in the Netherlands, we instituted a large project to figure out what exactly could be done with what the predictive value was of animal experiments. The use of animal studies in development pharmaceuticals. We also relied on old quote-unquote literature from Olsen, for instance, concordance of the toxicity of pharmaceuticals in humans and animals. A large study by 10 pharmaceutical companies. They put all the data together and they came to some very interesting conclusions. And one of them is that in many cases animals don't tell you the truth, which is good for them but bad for us. The second one is a little later. This is about not toxicity but efficacy. And the conclusion from this study is that concordance between animal studies and clinical studies varied for 6 interventions. 3 had similar outcomes and 3 did not, which is also what you get when you throw a coin in the air and look at what side comes up. So is that surprising? Basically it's not, because we look at inbred animals, very artificial disease models, and we assume that mankind is something like this, and we know that it is something like that. So it is not a surprise that when you look at animal work, caution is the key word. And here again, I think this is why CLINAM is so important, because CLINAM really makes the jump to the patient who is the major target there. So if you want to target the patient and not the drug. Having said that, I looked at the program and I also looked at some other CLINAM programs, and what I really miss is the this discussion, the discussion of what are animals trying to tell us and how much can we do with these animal experiments. You can fill journals, you can fill your pipeline of articles in PubMed, but does it really help the patient? And so over time we saw this EPR evidence or fallacy from Johan Bay from Utah, discussing and debating about this. Basically the message is do it as soon as you can in the clinic. And an interesting example of the first tries there, and I think that Dr. Dumont will talk about that later more in detail, and maybe the other speakers will discuss it as well. But this is Harrington's story that got, as far as I'm concerned, much too little attention, where he published in 2001 this study with 17 patients, and don't look at the details, different tumors, different solid tumors, and a very different percentage dose per kilogram taken up in the tumors, varying from nothing to 53% ID per kilogram. There may be some questions about the study, but the bottom line is that here, for the first time, as far as I know, somebody really tried to see who are the patients that really can benefit from the EPR effect. Okay, I'll move on to the next and last part of my presentation. Nanomedicines, complex drug products. We talk about complex drugs that are formulated in different ways, where the production process is very important in terms of the success, and the characterization is not so easy. When you look at the different examples given here, you see the liposomes, of course, mice cells, and dendrimers. But there is an interesting one. Here's antibody-drug conjugates. They are part of the nanomedicines family, apparently. And what I don't understand, why the most successful nanomedicines, molecular antibodies, are not part of this story. Why do you kick them out? What is wrong with them? Or what's wrong with you? Molecular antibodies, dimensions in the nano range, they're pretty large. And apparently when you make it a conjugate, then you count them, it's part of the family. They have an effect, clearly a therapeutic effect. Here, direct modulation of target antigen, complement-dependent cytotoxicity, antibody-dependent cellular cytotoxicity, and apoptosis. They do things, they are active. And with all due respect, what is nanomedicine? What does it mean in money-wise in the world? Nothing when you compare it with antibodies. Antibodies, 100 billion last year. That is success. So why am I talking about that? And then the targeting story. That's interesting, because molecular antibodies are detargeting, are Mother Nature at its best, her best. The detargeting moiety by excellence. Look at what happens. Impact of tumor volume and other characteristics on the uptake of radiolabeled monoclonal antibodies. What do they find? 27% ID per kilogram for different patients with solid tumors. That is for monoclonal antibodies, the best that there is. Now look at what Harrington found. 33% per kilogram, the same amount. And one is a big success therapeutically, and the other sort of struggling around. So my advice is also look at the molecular antibodies as a sort of example how successful things can be without extremely high targeting moments and effects. I see that I have 25 seconds left. Target accumulation between liposomes and molecular antibodies similar. This is another one of these publications on the same issue. And, well, of course, what it means is that we have to find the patients who really respond in advance. And already in 2011, Gert and I published something like that. But we were not the first ones at that time. I mean, it was sort of in the air at that time to combine the two. And that means, conclusions, drug target accumulation is not a goal in itself. Nanomedicines are successful because they favorably change the efficacy safety balance. Animal studies, caution. Maps, look at them as examples of highly successful nanomedicines. And of course, what we will discuss later today, this afternoon, personalized medicines, medicine through personalized medicines, time to integrate advanced non-invasive imaging approaches and smart drug delivery systems. With that, I think time is up, so Thanks for your attention, and I don't know whether it's time or necessity to talk. Questions?
Yes, thank you. First question.
Yeah, thank you very much for this very great talk.
Use the speaker. Yes.
Thank you very much for this great talk, and I think you came to a very similar conclusion like me. So why did the people of the nano field did not learn from antibodies? And I would even go one step further, and I will also address that point in my talk that comes next, is why did nature make antibodies as large as they are? And why does nature have antibodies of different sizes? Yeah, we have antibodies of 50 nanometers, we have antibodies of 35 nanometers. So why is that so? People always only argue about affinity and binding properties, but they don't really think about why are the sizes different. And I think we can learn a lot from these systems that are optimized for EPR-based accumulation, optimized for active targeting. Yeah? And I think we should find a link between the natural systems and the systems that we artificially design.
Well, I agree, of course, with what you're saying. The interesting part about the size of the monoclonal antibodies, that is indeed, why are they so big? Well, because they can circulate long and blah, blah, blah. But the gentleman sitting behind you will talk about nanobodies, I understand. So he will explain why Mother Nature is so stupid to make these large monoclonal antibodies, and why he is so clever to sort of think that he can beat Mother Nature.
Yes. Klaus Michael? Just shout.
Okay, yeah, no, good. Yeah, Daan, of course, very nice presentation, and I'm fully with you in that what you said. But the last year's paper and view, this whole discussion about the pessimism of nanomedicine, to my opinion, was of kind the tunnel view of those people working in the field of cancer and targeting. There are many other fields, and I try to give an example, for instance, infectious diseases, but that holds also for oral drug delivery, that holds for inhalation and topical delivery to the skin, where you can, and there are even products on the market of nanomedicines for oral administration, where they really have proven their superiority. Just if we reduced it to the problem of treat cancer and achieve targeting after parenteral administration, then you see these maybe not so convincing results. But there are so many areas, if we take nanomedicines as a whole field and not only talk about targeted cancer delivery, we cannot say that there is no success.
Well, I'm not going to argue about that. I heard your presentation, very good one, a lot of very promising things, but no products. And I'm counting on the products. But I really like what you were saying. I mean, there are other opportunities.
Yeah. I was actually less referring to what that is ongoing. This is hypothesis-driven, this is academic research. But when you look at these nanocrystals, these technologies which are based, and these are called nano and correctly called nano, these are medicines where you make small particles, take the advantage that you can improve solubility. This is a very modest, maybe low-hanging fruit, but there are products, and yes, that's nanomedicine, and we should not forget this.
I take that as an, yeah.
Gezi?
Well, Daan, today a method was developed actually that you can take your deoxyliposome. We're doing it now also with steroid liposome. We just add the zirconium to them in a special process. You don't need anything in the same liposome you can use to human or animal. We get fantastic results. So I think you can, for every patient, during the first patient treatment, you can say what will happen. And I think in a little while, the problem that not many places still use zirconium, because it has the right half-life to cover you for 3 days or so you can get a very nice information. Then, you know, I think if you look at patients, and I look a lot at patients and have a lot of contact with physicians, I think when we as a scientists come and talk about success or lack of success, we really don't know what's going on in the clinic. And you know, when seeing some patient that you treat with drug and nothing happened to them, then you give them a nano drug, and you can see the patient responding for many months and many years. That's a big success to me. And you cannot argue with success. You can argue with everything. I agree that the animal study teach you very little, and the problem is that we work with the wrong models, and that's the main thing. And we should find better models of spontaneous tumors or something more relevant to the human tumor, and then I think the animal will serve you very well. But we are too lazy to do it. It's very complex to do. So then we show success in these very nice models that works very nicely, but then when you come to the real situation, it's irrelevant, the model. So I think we have today many more answers than we have before. We have to apply them. I think in 5 years, we have much more information on the system that can tell us what to do. We also have a way to overcome major obstacle of these nanodrugs, because the long circulation does have also a downside implication that we have to take care of, and different types for different drugs, but I think it can be handled. And actually, we have good proof in patients now that we can overcome completely complement activation, and we're developing now into something new. So I believe you can deal with this question, and really it open and improve the way you treat. And the last thing, which I think we all talk about the EPR effect as the holy grail. I mean, but you can get to a situation that you get terrific accumulation of drug in an excellent EPR effect in patient, and you get zero therapy because the drug does not release. It stays there like stones in the liposome and nothing happens. So I think the bottom line is a combination of EPR effect, but more so what happened to the release and what kind of release we want. Do we want a fast release? Fast release has maybe a good chance of success, but also it will get we get development of resistance, or we want a slow release below the level that causes resistance. All these questions were not answered yet. So we have a long way to go, a very long way to go. And I think the point is that people are jumping from one issue to the other and not concentrated on one system, really try to solve it. And this will be later a lesson to all other systems to be used.
Next time, Gezi, you will speak before me, and I will speak after you. Because I think you make a lot of points that are very valuable, but there is no question, I think. So in that sense, I leave it for what it is, but 80% I agree with you. And of course, the rest we have to fight out outside. Yeah, usually it's less, I agree with that.
Good. So thanks to both for the discussion. Thanks, Daan, again for a very nice talk.